My name is Henning Ulrich, and I would like to start our symposium today on Purinergic signaling. I would like to announce the program of our symposium in the afternoon. It's a symposium on Purinergic signaling, and this symposium will be held in the memory of Professor Jeffrey Bernstock, who is the father of Purinergic signaling. Some of you know him, and he has passed away recently. And in his honor, we will have a symposium which will be held by the speakers, Professor Rodrigo Cunha, the University of Coimbra, Portugal, and then followed by Professor Mariusz Ratacjzak, University of Louisville, and Professor Robson Coutinho Silva of the Federal University of Rio de Janeiro, and finally by myself. And I would like to thank the FeSBE and the 𝒂𝒓𝒊𝒏𝒂𝒄𝒋𝒛 for organizing this workshop. So now I would like to present our first speaker, Professor Rodrigo Cunha. He is an honorary member of 𝒂𝒓𝒊𝒏𝒂𝒄𝒋𝒛. He is a professor of the Faculty of Medicine at the University of Coimbra and principal investigator at the Center for Neuroscience and Cell Biology at this university. Professor Cunha is a leader in Purinergic signaling. He is working on neuromodulation of adenosine and ATP, and he has a great input, especially in the understanding of neurodegenerative diseases, such as, for example, Alzheimer's disease and also on aging. So I would like to invite Professor Cunha to give his speech, and we are very much honored by your presence and contribution to this symposium. Professor Cunha, thank you very much.
So first of all, good morning, everybody. I hope you're hearing me in good conditions, and you are seeing the first projection, which displays the title of my presentation, which will focus on aging. So it is an overarching topic within the University of Coimbra. Here you see the new building of the Multidisciplinary Institute of Aging, which is actually recruiting researchers as I speak, and will continue to do so in the next 2 years, with a total of 60 new researchers to be hired in this multidisciplinary institute. So when I talk about purines, most of you may get a little bit lost. It's actually something that you know very well and you do on a regular basis to manipulate this system. And you do so essentially by drinking something that a lot of you look at with some suspicion, which is coffee. This is normally associated with increased anxiety, and very commonly doctors, when they foresee some problems in individuals, they recommend avoiding the triad of major toxins in their mind, which is ethanol, nicotine in the form of smoke, and coffee goes along this list. So what I will try to do in the very first part of this presentation is to challenge this idea and to provide you data that may force you to look at coffee differently as something beneficial to your health, and actually something that, without you knowing, has a large impact in a lot of functions within your bodies. So linking directly coffee with aging, one can ask whether or not people that drink coffee on a regular basis will have a better process of aging. And this is one study which was published in a prestigious medical journal, showing and studying the association of coffee with mortality in general. And what you see in here is a line. Towards your right-hand side, things get worse. Towards your left-hand side, things get better. And when you look at the vast majority of the problems that can occur on aging, for most of them, there's a pretty good evidence, accumulated evidence, that there's a benefit associated with the intake of coffee. This is just not one study. It's actually a rather robust finding with several cohorts showing exactly the same data in Europe, in America, in Asia, with those response curves rather well established, with effects that are more evident as we engage in the late years of our life, and valid for different types of coffee, valid for men and women, for different ethnicities and for different phenotypes, that is, different polymorphisms. If we are fast or slow metabolizers, alike, we get a benefit. So there's a lot of body systems where this benefit is well established. This has been recently reviewed in a paper from about 2 months ago, published in the New England Journal of Medicine. Again, this is day drawing, this is my drawing, basically to let you know that in different individual body organs, you do have a benefit. For instance, if you look at the liver, the incidence of fibrosis, cirrhosis, or hepatocarcinoma is lower in people that drink coffee, and so on. There are general benefits, clear benefits in diabetes, in obesity. Different types of cancer actually have a lower incidence in people drinking coffee, as well as on basal inflammation. And this is not, just all benefits. There are question marks still. When it comes to osteoporosis, more studies need to be done. There are still some open questions about pregnancy, that need to be addressed. Some people, a small fraction of people, are affected in terms of modification of their sleep pattern, and the GI side effects have been largely ruled out as a counterindication for coffee intake. Now, what in coffee? Why is coffee affording such benefits? Now, one thing we can say is that this is due to other things apart from coffee. Normally, you go and take a coffee with somebody, you're relaxed, and it could be the social part, the social interaction, or the relaxing, which may have some benefits. It is difficult, actually, to study the impact of coffee intake, since it's widely available to all the population. So you need to ask people to stop drinking coffee, and it doesn't make it easy for coffee drinkers to be part of their favorite drink. Obviously, there is not yet a mechanistic basis for the reason why coffee affords such benefits. There's one good candidate, which is a main component of coffee, which is caffeine. And this is where coffee meets the purinergic system. It's because caffeine acts on the purinergic system, as we will see. Most of the information I will present you from now on has been obtained in animal models, which are critical, especially to overcome this critical aspect of availability of coffee and controlled administration of coffee or coffee components. So we've been looking in animal models at the impact of, in particular, of caffeine, and sometimes comparing it with that of coffee extracts. Now, if you think about caffeine, what are the molecular targets of caffeine in the human body? So essentially, and in contrast to what is still stated in most pharmacology textbooks, the only target for caffeine at non-toxic doses is the antagonism of the two main adenosine receptors. What pharmacology textbooks state is obviously correct. Caffeine has other targets, but it targets those targets when we are essentially in pretty bad shape, that is, in toxic doses of caffeine. Those adenosine receptors, which are main components of the purinergic system, where are they located? Well, as you can see, they are mostly present in the brain. They are spread throughout the brain. And then you can ask, in which conditions do you generate the purine adenosine, which activates those receptors? And this does happen in the brain under two different conditions. When you increase the activity of brain networks through their electrical stimulation, you get an activity-dependent release of adenosine. And adenosine is not a neurotransmitter, but it is instead a neuromodulator. That is, it modulates ongoing activity, it fine-tunes ongoing activity. If you now apply noxious stimuli, such as this case, displayed hypoxia, you do also, produce the release of adenosine in far greater amounts, and this is associated with the role of adenosine as a danger signal. As a danger signal because every time there's an increased workload or there's a need to repair within any cell, cells use ATP, and the A of ATP, it's the adenosine. And since cells are endowed with bidirectional and non-concentrative nucleoside transporters, they increase extracellular levels of adenosine as a function of the formation of adenosine within the cell according to the workload or stress encountered by the cell. So anytime there's increased workload or stress, what you get is an increased amount of adenosine. And this adenosine is important to control the demise of degeneration and of neuroprotection within the brain tissue. So very rapidly, and summarizing something like 20 years of experimental activity, the role of adenosine is essentially to encode salience of information within neuronal networks. So normally, when neuronal networks are just maintaining activity, they are not retaining novel information, there is an ongoing activity, an ongoing usage of ATP, an ongoing production of adenosine, because it's expensive to maintain the nervous system ready to work. We use about 20% of our body glucose just to maintain the nervous system ready to work, not working, ready to work. And this adenosine activates inhibitory A1 receptors, which are more abundant in glutamatergic synapses, excitatory synapses. And this is like a low pass filter whereby you decrease noise, you decrease activity in all excitatory synapses. Now, when you need to encode information within networks, you do so by changing the efficiency of synapses. In here, it can be coincidences of signal arrival. It could be frequency. Here is displayed frequency triggers. And as you need to retain information, now you release a far greater amount of neurotransmitters. Now the amount of ATP which is co-stored with neurotransmitters in vesicles released within the synapse increases. And when this increases, there's a cascade of enzymes that convert extracellular ATP and extracellular adenosine. And this other receptor, the facilitatory A2A receptor, is physically coupled to those enzymes and is activated at the mouth of where adenosine is produced. When it is engaged, it shuts down the A1 receptor presynaptically, bolsters the release of more neurotransmitters, and activates the trigger of plasticity, which is a ionotropic receptor for glutamate of the NMDA subtype. What is triggering plasticity, actually? So in this synapse, what you have is increased release of transmitters, you block facilitations, you increase the trigger, more glutamate activates the astrocytic syncytium, and in all synapses covered by these syncytiums, what you have is more adenosine because greater activity is imposed on those cells. And more adenosine means greater inhibition in all neighboring synapses caused by this system. So what you have is greater facilitation in the activated synapse and greater inhibition elsewhere. So what you're doing is to increase salience of the signal being encoded. Now shifting to aging. In aging, this goes wrong, and you have a shift of the receptors. You have, first of all, a decrease in the controller of noise, so more noise is encoded, and at the same time, you get an increase of this facilitator of encoding to levels that get dangerous to cell. And this is corrected by caffeine, which prevents this excessive function of A2A receptors and normalizes the ratio of functioning of the two receptors. Let me show you some of the evidence we've collected for that. So this is what happens as rats age in terms of density of the A1 receptors. You can see that as the animals become old, you see that there's a progressive reduction of the density of A1 receptors, which is accompanied by a decreased expression of A1 receptor mRNA. Now, when you look, this is in the hippocampus, I forgot to say, a brain region related with memory performance, amongst other things. Now, when you look at the other receptor, the A2A receptor, you see precisely the opposite. You see an increased density of the A2A receptors. You see that this is particularly prominent at the time of aging, rather than slowly progressing as the animals age. And this is also accompanied by an increased expression of the A2A receptors. Now, this is true in rodents. It's also true in humans, as shown here very rapidly. You see increased age of volunteers with at least no neurological disorders at the time of autopsy. And what you see is that there's an increased density of A2A receptors, in this case in the cingulate cortex, as the individuals age. Now, this increase in density of the adenosine A2A receptor is even greater when there are diseases associated with aging, such as in this case displayed here, Alzheimer's disease, patient diagnosed with Alzheimer's disease at autopsy at levels of A2A receptors in the hippocampus far greater than those of age-matched individuals, which were far greater than young individuals. And in general, this shift from a predominant inhibition to a predominant facilitation, this overactivation of A2A receptors, is in a way a process that prones for disaster, increases the susceptibility to neurodegeneration to occur. Now, we tested that in the case of aging, because in aging, you already have this shift. It was also known that you have behavioral modifications, namely of memory, in a large proportion of animals. So we wanted to know, if we block this excessive function of A2A receptors, can we actually reverse the age phenotype, in particular the memory phenotype that occurs on aging? So what we did was to compare the impact of blocking A2A receptors with a daily injection during 3 weeks of a selective antagonist of this receptor. And we tested that in young adult animals and compared it with aged rats. The data I'm going to show you is only of males, and we have now completed the studies showing that the same thing occurs with females, of what I'm about to show you. So the first thing we did was a behavioral characterization of the animals. And as displayed here, you don't have a change, a global change in locomotion. You have a decrease of sensory motor coordination assessed by the number of rearings of the animals in an open field test, which is not corrected by blocking A2A receptors. When you now consider anxiety, there is no real trend for increased anxiety in the aged animal, be it in the time exploring the central part of the arena, which is more aversive to the animal, or to the time they spend in the more aversive open arms of an elevated plus maze. Now, in contrast to this lack of motor and mood-related modifications, they are deficits when we investigate reference memory. So in this modified Y maze, where we hide one of the arms, and the animal has to remember, according to spatial cues, that he was not yet, did not yet have the opportunity to visit this new arm. So when it is available, they spend greater time than chance searching for it. And this is so for the young animals, but the aged animals do not remember that, do not seem to remember that they did not previously visit this arm. And as you can see, this is prevented if the animals are treated for 3 weeks with the A2A receptor antagonist. In the object displacement test, similar rationale, an object is displaced. It's an hippocampal dependent test. As the object is displaced, the animal tends to go there more to search for this object, more than chance, as shown in the young animals. This does not occur in the majority. I'm talking about 90% of the aged animals that we test, and we recover that when we block the A2A receptors. We try to confirm this deficit of reference memory and this ability of A2A receptors to correct it by looking at another test. And we looked at the Morris water maze. Learning is not so effective. They do learn, but they take longer. In the aged animals, this is corrected by the A2A antagonist. When we test for the memory of the animals in the probe test, they take more time to get to where the platform should be, spend less time there. This is corrected by blocking the A2A receptor. There's no change in the swimming speed, motor function. In the visual test, there's no differences. In the reversal learning, when we change the rules, now the platform is no longer there, but you have to learn that it's elsewhere. Again, they show deficits corrected by blocking the A2A receptors. And this is a campaign very rapidly by the loss of synaptic markers, in particular of glutamatergic markers, which remarkably are recovered by blocking the A2A receptors. A lower synaptic plasticity, in this case only displayed long-term potentiation in the aged animals, which is recovered in terms of amplitude in the hippocampus C1 area by blocking the A2A receptors so rapidly. In conclusions, as coffee has a benefit for elderly individuals, we now show that this is also true in animals selectively by targeting the A2A receptors. We have looked so far only at memory. This beneficial effect of A2A receptors is particularly striking when you look at Kaplan survival curves, showing that you have an increased survival in animals where you eliminate the A2A receptors. We don't know the targets. We follow Felipe Sierra, candidates for senescence. Caffeine interacts with them all, and we want to try and dissect which one is particularly affected by A2A receptors in the brain. Finally, thanking all the collaborators throughout the years and the funding agencies that made this possible. And Henning, I turn down to you because I've concluded my presentation. Thank you very much.
Rodrigo, thank you very much for this impressive talk, which we will discuss later on. Now, I would like to present our second speaker, Professor Mariusz Ratacjzak. Professor Mariusz is also a member of the Brazilian Purin Club. He's professor and endowed chair of the Stem Cell Institute at James Graham Brown Cancer Center at the University of Louisville. And he is a very well-known specialist in the field of adult stem cells. And he has discovered the embryonic-like stem cells, small stem cells. And he has also a huge expertise in the developmental biology aspects of stem cells, and he is a hematologist. He's working with complement and homing, and lately, a couple of years ago, he has joined the field of purinergic signaling, and since then done very important contributions to the field. Professor Ratacjzak, thank you very much for contributing to the symposium and sharing your expertise.
I would like to thank very much, for invitation to this great meeting, and Dr. Henning Ulrich for this very kind introduction. And we will switch right now from neurology to hematology and purinergic signaling. And first of all, hematopoietic stem cells are unarrested travelers. Why? Because they traffic through our body during development. They have some circadian rhythm of circulation. More of the stem cells is in morning hours than at night, and I will show you purinergic signaling plays here an important role. Number of the cells in peripheral blood increases after strenuous exercise, tissue organ injury, but what we will focus today, after pharmacological mobilization, if you give to the patient granulocyte colony stimulating factor, cytokine GCSF, or 𝑥cr4 alpha-chemokine receptor blocker MD 3100, you can increase number of these cells in peripheral blood up to 100 times. And this is right now a mean to collect stem cells for hematopoietic transplant. This means instead of isolating these cells directly from bone marrow, we can isolate these cells from circulating blood by plasma leukapheresis. And then these cells could be again transplanted. This means infused intravenously to the patient who first is undergoing preparative regimen by total body irradiation or intensive chemotherapy to kill old hematopoiesis, and transplanted intravenously stem cells from the donor, like from mobilization, they engraft in the bone marrow. They have also traveled from peripheral blood to bone marrow and graft and establish hematopoiesis. But both, mobilization and in greater extent conditioning for transplant by radiochemotherapy induces sterile inflammation in the bone marrow. This means sterile inflammation which is not caused by microbial infection, but some activity of innate immunity. First, before we will move further, stem cells, as is shown here on the scheme, are retained in the bone marrow, hematopoietic stem cells in niches, like a bird in the nest. As you can see here, 𝑥cr4 receptor, which is interacting with alpha-chemokine stromal-derived factor secreted in bone marrow niche. And what is here very important, what I will show you a few times in today's lecture, 𝑥cr4 is membrane lipid raft associated receptors. Membrane lipid rafts are special areas in membrane enriched in sphingolipid and cholesterol. And as you can see here in evidence, confocal microscopy is human hematopoietic stem cells, and we have stained these cells for lipid raft using here special fluorochrome and antibody, and also for 𝑥cr4 receptor, and confocal microscopy provides evidence 𝑥cr4 colocalized with membrane lipid rafts. Why it is so important? Because SDF1 stromal derived factor signaling through 𝑥cr4 is much more effective and provides much stronger signaling if 𝑥cr4 is included in membrane lipid rafts. And this process, on one hand, could be stimulated by some cationic antimicrobial peptides, which are listed here on the scheme, and then cells much better respond to SDF1 gradients, are hungry for this gradient, much better migrate to bone marrow, much better engraft. And what I will be showing you today, extracellular ATP is one of these very important factors which promotes inclusion of 𝑥cr4 into membrane lipid rafts on hematopoietic stem cells. Now purinergic signaling in response to bone marrow stressors. First of all, ATP as danger-associated molecular pattern molecule is very important because it's released in bone marrow in response to agents which promote mobilization like GCSF and MD3100, but also during conditioning for transplantation by radiochemotherapy. And mostly it is secreted in Pannexin 1 channel dependent manner. Of course, ATP is processed by ectonucleotidases CD39, CD73 to adenosine. It is why ATP and adenosine seem so very important in stem cell trafficking, and I will show you that they have yin-yang effect on stem cell trafficking. This means positive and negative. Positive by ATP, negative by adenosine. And to address this question, we have phenotype here, human bone marrow hematopoietic stem cells and mononuclear cells by PCR, looking for different P1 and P2X receptor. And we found that among P2X receptors, in particular, is very high expression of P2X4 and P2X7. In terms of P1 receptor, is very high expression of A2A and A2B receptors. And now, this receptors has connection to inflammasome. I will be talking more about inflammasome. It is very important part of innate immunity signaling machinery expressed in innate immunity cells, granulocytes, monocytes, macrophages, dendritic cells, but also what we recently reported in hematopoietic stem cells as well. And this inflammasome is activated by danger-associated molecular pattern molecules, including ATP, and it is causing sterile inflammation of bone marrow. You can see here our data showing that LLP3 inflammasome components are expressed in human hematopoietic stem cells and also in murine hematopoietic stem cells. And what we believe here, purinergic signaling by ATP and activating inflammasome, NLRP3 inflammasome, and NLRP3 inflammasome is a kind of cogwheel which connects purinergic signaling with complement cascade activation and induces sterile inflammation in bone marrow. Here is maybe more detailed scheme how it happens. ATP activates P2X7 or P2X4 receptor. I selected these receptors because they are very highly expressed on hematopoietic stem cells. And the activation of this complex activate caspase-1, and several factors are released from activated cells, including interleukin-1 beta, 18, but also some other molecules I call a molecular box group protein 1. Now coming to hematopoietic stem cell mobilization, this means egress of stem cells out of bone marrow. What I will be showing you here, after administration of promobilizing agents like GCSF or MD3100, cells are activated, release ATP by Pannexin 1 channel, and then through P2X7 receptor, this means P2X4 and P2X7, we have activation of NLRP3 inflammasome. And here's some data. As you can see here, mobilization by GCSF and MD3100, and as you can see here, it is increase in ATP in peripheral blood of mobilized animals, which could be inhibited by probenecid, one of inhibitors of Pannexin 1 channel. Of course, we repeated this right now with Pannexin blocking peptide. But if we add to mobilizing protocol by MD or GCSF ATP, as you can see, we can increase number of mobilized hematopoietic stem cells into peripheral blood. And we can inhibit this effect if mice are exposed to probenecid or Pannexin 1 blocking peptide. Here are data coming to receptors activated by ATP, P2X7, P2X4. As you can see here, in case of P2X7 knockout mice, we have observed that these mice are poor mobilizers with GCSF in comparison to wild-type animals, both in 3-day mobilization, 6-day mobilization protocol. But somehow we were surprised it was no difference in response to MD3100 in these mice. But then we performed very similar experiments in P2X4 knockout animals. Again, P2X4 knockout animals have decreased in mobilization of hematopoietic stem cells as compared to wild-type animals after GCSF, but in contrast to P2X7 knockout, also it is effect of MD3100. Not coming right now more in details, we believe that difference is due to a fact that P2X4 is much more potent and much faster signaling receptors than P2X7. This means in 1 hour mobilization with MD3100, we see already effect in P2X4, but not yet in P2X7 because it is delayed effect. Then we went to NLRP inflammasome knockout mice, and we found that these mice, and as you remember in my scheme, I mentioned NLRP3 is downstream of P2X4 and P2X7. These mice are poor mobilizers. This means lack of NLRP3 causes poor mobilization in wild-type animals, both after GCSF and MD. And here in this, to this part of my presentation, our already published data shows that in GCSF MD3100, very important role are playing several here elements of innate immunity and purinergic signaling. Everywhere there are asterisks, it is defect in mobilization. This means lack of innate immunity cells, leukocytes, granulocytes, dendritic cells is poor mobilization. Lack of panexin-1 functional channel is poor mobilization. A lack of P2X4, P2X7, poor mobilization. NLRP3 and caspase-1 knockout mice we have already published, poor mobilization, and also defective activation of complement cascade. Just very briefly, as I mentioned, it's circadian rhythm of hematopoietic stem cell trafficking in peripheral blood. More of these cells are in morning hours, less at late night. And here are our data showing fluctuation in ATP level in peripheral blood of mice, and highest level is at approximately at 11 at noon. And here it corresponds with number of circulating clonogenic progenitor stem cells in peripheral blood, which is the highest at 11 a.m. And also it is inhibited by panexin blocking peptides. This means here ATP has to be released in panexin dependent manner, and also by inhibitor of NLRP3 inflammasome MCC 950. Now, another part of my presentation, reverse process to mobilization, reverse process to egress. This means homing and engraftment in the bone marrow after transplantation. And the most important factor here, which attracts stem cells to bone marrow conditioned by lethal irradiation or chemotherapy, is stromal derived factor 𝒂𝒂𝒂𝒄. But also some role is playing bioactive sphingolipid sphingosine-1 phosphate, and also ATP released in irradiated or damaged by cytostatic bone marrow microenvironment. And again, this activates NLRP3 inflammasome. Here, as you can see, here is very nice evidence that if we expose cells to SDF, SDF is very strong chemoattractant, control SDF of hematopoietic progenitors. But this effect is inhibited in presence of panexin and also in presence of aperase, also for other chemoattractants. What is here important implication from this? That, you know, somehow SDF cooperates with ATP probably secreted from stem cells in autocrine manner to promote this chemotaxis to SDF1. And here are also our already published data. As you can see here, inhibition of Panexin-1 channel in vivo in mice by Panexin blocking peptide releases and inhibits their homing and engraftment. This means ATP has to be released by stem cells that they sense much better as SDF1 gradient, because as exposed to Panexin blocking peptide, as you can see here, is much less of stem cells which engraft in bone marrow 24 hours later, and also as evaluated here 2 weeks after transplant, and also is difference in hematopoietic reconstitution, looking for number of white blood cells and platelets. And here is a response to this. Here are normal lipid rafts on hematopoietic stem cells isolated from mice, but if we expose the cells to Panexin blocking peptide, we don't see lipid rafts. This means here is our model showing the hematopoietic stem cells respond to SDF1 and also other chemoattractants released in bone marrow during engraftment and homing, but also ATP, which is released from cells where NLRP3 is activated by SDF1 in autocrine manner, promoting inclusion of homing receptors, in this case CXCR4, into membrane lipid rafts. And here more data. Here are mice which are exposed to Panexin blocking peptide and then engrafted with wild type bone marrow. And as you can see here, early homing and engraftment was impaired when Panexin-1 channel was blocked in hematopoietic microenvironment of recipient mice. Here is inhibition of P2X4 receptor on hematopoietic stem cells before transplantation. Cells before they were transplanted, we inhibited P2X receptor by specific inhibitor. And again, early homing and early engraftment and reconstitution is defective, showing that this receptor has to signal properly. Here is reverse a little bit experiment. We have exposed recipient mice to P2X for inhibitor, and as you can see here, again impaired homing and engraftment of hematopoietic cells. And here, you know, P2X7 receptor was affected by hematopoietic cells, and again, defect in engraftment, early, late, and hematopoietic reconstitution. And here's the same with P2X7 receptor, which was perturbed in hematopoietic microenvironment. Here also, to summarize this, both receptors are important to be properly expressed on hematopoietic stem cells while they migrate to the bone marrow. On other hand, these receptors have properly signal in bone marrow microenvironment in homing and engraftment. And here is shown how important in this is NLRP3 inflammasome, because if we employed here knockout mice, NLRP3 knockout mice, which don't activate NLRP3 inflammasome is of course not activated properly, regardless if P2X4 and P2X7 is expressed, these mice have defect in early engraftment, late engraftment, and also in hematopoietic reconstitution after transplant. And here is also an evidence that, you know, have really NLRP3 is very important in migration, because here in chemotactic transfer assay, as you can see here, cells from this knockout mice much worse respond to SDF1 and other chemoattractants playing a role in homing. And here is also verification in this, you know, NLRP3 white type mice were engrafted, had defect in engraftment in wild type bone marrow. And this is again explained by effect of extracellular ATP, P2X4, P2X7, and NLRP3 signaling information on membrane lipid rafts. Because as you can see here, again, hematopoietic stem cells have proper lipid rafts in response to SDF1. But if we expose them to uparase, which actually destroy extracellular ATP, we don't have lipid raft formation. Now, what inhibits this process is adenosine hemooxygenase 1 axis. We are working on this. You know, we have already reported in the past that hemooxygenase 1 is inhibitor, negative regulator of homing and mobilization of stem cells. There are two different, most important hemooxygenases, 1 and 2, but hemooxygenase 1 is just expressed, is induced during stress, and it's inducible hemooxygenase. And as you can see here, if we have exposed hematopoietic progenitors in transfer migration system to adenosine, we observe decrease in migration. Here is also in vivo experiment. Mice were injected with still tolerable dose of adenosine, and as you can see, they have shown defect in mobilization by GCSF and AMD. And here you can see that, you know, if we expose these mice to adenosine, to ATP or adenosine, in case of ATP, ATP downregulates hemooxygenase 1, adenosine upregulates hemooxygenase 1 expression. It is why we are coming to this here conclusion that trafficking of hematopoietic stem cells is regulated by proper purinergic signaling P2X7 and P2X4 receptor activating NLRP3. But ATP metabolite adenosine 1 by upregulating hemooxygenase 1 has opposite effect by inhibition of inflammasome. And there are several reports, and also our report directed in hematopoietic stem cells, that hemooxygenase 1 is inhibitor of NLRP3. And here, you know, also few data with the membrane lipid rafts. As you can see, NLRP3 knockout mice, in contrast to wild-type mice, have defect in lipid raft formation. But also we can inhibit lipid raft in wild-type cells by exposing to adenosine or upregulating adenosine by upregulating of hemooxygenase 1 by one of the stimulators of hemooxygenase 1, which is COPP. And in this final slide here, how we envision navigation of hematopoietic stem cells to most important SDF1 gradient. SDF1 by activating ROS, it activates inflammasome. But also at the same time, paracrine ATP is doing the same, activating P2X7, P2X4, which activate inflammasome. This releases ATP, which promotes incorporation of CXCR4 receptor into membrane lipid rafts. And this happened everything and leading age of migrating hematopoietic stem cells. But then this ATP is degraded, metabolized by CD73 and CD79 ectonucleotidases to extracellular adenosine. And based on our data, because A2A and A2B receptors are very highly expressed on hematopoietic stem cells, this signal, we still don't know which here of this receptor is most important. And we are working on this and count here on collaboration with Dr. Cunha, you know, to have a proper knockout mice to address which of these receptors is important in upregulation of hemooxygenase 1, then to inhibit inflammasome, then to inhibit cell migration. Conclusion from my presentation that extracellular nucleotides are important regulators of trafficking of normal hematopoietic stem cells. And extracellular ATP released from the cells into extracellular space activates by two receptors, P2X7, P2X4, and NLRP3 inflammasome, which is very important here in migration of hematopoietic stem cells, because the deficiency of all these elements results in pull mobilization, pull homing, and engraftment. And while ATP activates NLRP3 inflammasome and stimulates migration, its metabolite, extracellular adenosine, has an opposite effect. And we are working right now to dissect which of the adenosine receptors plays here a crucial role. And then to take home message, especially for hematologists, that stimulation of ATP and NLRP3 inflammasome axis, for example, by potent stimulators of P2X4 or P2X7, may improve mobilization. We can get more cells for transplant from the patient. And vice versa. Here, inhibition of extracellular adenosine hemooxygenase 1 signaling can improve engraftment and homing. And I would like to thank very much here my people who are working very hard, especially Mariusz Adamiak, who right now make his habilitation on these projects, and also other people working in my laboratory, and most important, my collaborators, Professor Henning Ulrich from University of São Paulo, Dr. Ratacjzak, Dr. Magdalena Cunha, and also Dr. 𝒂𝒂𝒊𝒊𝒉𝒂 from University of Kentucky.
I would like now to invite our third speaker. This is Professor Robson Coutinho Silva. Professor Robson is at the Institute of Biophysics, Chagas Filho at the University of Rio de Janeiro. Professor Robson is one of the founders of the Purinergic Purinergic Signaling Club, 𝒂𝒂𝒊𝒊𝒏𝒂𝒄𝒂. He is one of the most prominent researchers on purinergic signaling in Brazil, and basically he works on immunology, inflammation, immunomodulation, and also infection, yes? And he's a big specialist on this. And Professor Robson, he has been president of the Brazilian Purin Club, he has been vice president, he is a counselor of the Purin Club, and he has an incredible participation in establishing the Brazilian Purin Club, and also in fostering the science of purinergic signaling in Brazil and also abroad. Robson, thank you very much for accepting to give a talk in this symposium.
First of all, I would like to thank Henning for this kind introduction and for the invitation, and to Clube Brasileiro de Purinas for organizing this symposium. And today I'm talking a little bit about the interplay between purinergic receptors in response against leishmania infection. So, as Mariusz has shown to us, there are different situations, inflammatory situations, such as cell death by necrosis, apoptosis, or during immune response. We have secretion of ATP. And once in the extracellular medium, ATP can activate two families of purinergic receptors, P2Y receptors and P2X receptors. P2Y receptors are called 𝒌 receptors, and P2X receptors are ion channel, intrinsic ion channels. P2Y receptors and P2X receptors can be found in the majority of immune cells. During this talk, I will present some data from the function or the role of P2Y2 receptors, the high affinity receptor for UTP and ATP, and some data from the role of P2X7 receptors, low affinity receptor for ATP. So, I can skip from these slides, because have been introduced by Mariusz, but basically 𝒂𝒂𝒃 is seen as a secondary important component for full activation of inflammasome, leading to release of pro-IL-1 beta and IL-18 cytokines, especially in microarthritis immune cells. So, but we are talking about one disease, it's leishmaniasis. The leishmaniasis is a group of diseases caused by protozoan parasites. There are more than 20 leishmania species. They are transmitted by human, by bites of infected female phlebotomy sandfly. And there are 4 clinical manifestations of the disease: cutaneous, diffuse cutaneous, mucocutaneous, and visceral. We are talking about Leishmania amazonensis. Mainly, Leishmania amazonensis induce this form of the disease, it's cutaneous one. If we consider the global distribution of leishmaniasis, we can see that we can find this disease in all 5 continents. And 1.5 million new cases occur each year. In particular, in South America, you can see that it's a strong endemic area. So it's a health problem here. So we start work in this field 11 years ago when Susanna observed that ATP treatment can reduce the parasite loads, here shown using GFP transfected fluorescent leishmania. So you can see that in a dose-dependent way, ATP can reduce the parasite loads. And also, after infection, macrophage infection, we see that this infection can increase the expression of 𝒂𝒃7 receptors. Then the direct correlation with 𝒂𝒃7 was shown when we infect macrophage from 𝒂𝒃7 knockout mice. And you see here that ATP do not have any effect in reduction of parasite loads, different from macrophage from wild type, and quantifier here. So when we infect the animals, 𝒂𝒃7 knockout animals, and follow the development of the disease, we can see that the lesion grows faster and are bigger in 𝒂𝒃7 knockout mice than wild type. And when we euthanize the animals, it's easy to see that we obtain more parasitic load in 𝒂𝒃7 knockout mice and cellularity increase. Also, the profile of the cytokine is completely different. In knockout mice, we have increase in interferon gamma and IL-10, but decrease of IL-12, IL-17, IL-4, and TGF-beta. But what would be the mechanism involved in the reduction of parasite load mediated by this receptor, 𝒂𝒃7 receptor? Well, at the literature, there are evidence that both nitric oxide and superoxide are involved in controlling of Leishmania amazonensis. So we checked if ATP can induce production of ROS. And as we can see here, in both uninfected and infected macrophage, we can see that ATP can induce ROS production. But we could not detect any production of nitrates, suggesting that ROS is the way how ATP is working or operating during the infection. But again, and how ROS can modulate anti-Leishmania effect. To try to understand this or to find the answer for this question, we looked for the literature and we found that lipid mediators, leukotriene B4, are important to control Leishmania amazonensis. And also, leukotriene B4 is associated with ROS. Then we evaluated if ATP can induce release of LTB4 in infected and uninfected animals, macrophage. And as we can see here, in both an uninfected and infect macrophage from 𝒊𝒏𝒈, ATP is able to induce release of 𝒂𝒐𝒏4. The same can be seen when we infect macrophage from C57-BLAK6. We have in uninfected and infected release of 𝒂𝒐𝒏4, but we could not see any release in macrophage from P2X7 knockout mice. To make short this first part of this talk, we see that activation of P2X7 can involve PLA activation, 5-lipoxygenase production of 𝒂𝒐𝒏4 and secretion of 𝒂𝒐𝒏4, and via PLT1 receptors, we have the effect of parasite elimination, parasite control. We also follow the pathway related to inflammasome activation. And as we can see here, wait a second, as we can see here, ATP treatment wild type can reduce the parasite control with ATP in wild type, macrophage from wild type, but we cannot see any reduction when we use macrophage from NLRP3 animals or ASC animals knockouts or double caspase 1 and 11 knockouts. And neither we can see reduction of parasite control when we have macrophage from IL-1 receptors. Also, in terms of the 𝒂𝒐𝒏4, again, 𝒂𝒐𝒏4 can reduce the parasite control, but we cannot see 𝒂𝒐𝒏4 reducing parasite control in macrophage from NLRP3 knockouts, macrophage from ASC, caspase 1-11, and macrophage from IL-1 receptors knockout. Then we check the importance of IL-1 receptors. To do that, we directly use IL-1 beta. And the treatment with IL-1 beta can induce the parasite load, but also can induce the parasite reduction in parasite load in macrophage from P2X7 knockout mice, from ASC knockout mice, and it's not possible to see any reduction in macrophage from IL-1 receptor. So suggesting that IL-1 is operating by its receptor direct. It's the same for 𝒂𝒐𝒏4. In this case, we checked if the non-canonical NLRP3 inflammasome could be involved. This means that if caspase 11 could be involved in ATP and 𝒂𝒐𝒏4 effects. And as you can see here, we have the reduction of parasite load in wild type, but we cannot see any reduction when you use macrophage from caspase 11 knockout mice. The same is for ATP, while in wild type, macrophage from wild type, we can clear here see the reduction of parasite load. In caspase 11, you don't see any reduction, suggesting that caspase 1 is important for the control for 𝒂𝒐𝒏1, 𝒂𝒐𝒏4, and ATP effects. Also, when we use an antagonist of caspase 1 and caspase 11, both are able to block the ATP effect. So again, suggesting that the pathway is important, and both caspase 1 and caspase 11 contribute to effect of ATP. So to make short this part, we say that the ATP can activate P2X7, producing 𝒂𝒐𝒏4, and the 𝒂𝒐𝒏4 by its receptor can activate caspase 11, that in the form non-canonic can activate NLRP3, caspase 1, release of caspase 1, active caspase 1, and by its receptor control the parasite. But what can we say about other purine receptors expressed in macrophage? So Camila has shown that UDP and UTP can also reduce the parasite load, and in a way dependent of intracellular calcium. At that time, Camila also shown that during the infection, we have in a mRNA level and a protein level increased expression of P2Y2. Later, Maria Luisa have proved the involvement of P2Y2 using a selective antagonist of P2Y2. She has shown that she can block the pre-treatment with a selective antagonist of P2Y2 can block completely the UTP and the ATP effects. Also, the phospholipase C inhibitor of phospholipase C can block the UTP and the ATP effect. So, proving that P2Y2 is responsible for the effect observed by UTP and ATP. But one unexpected result was that the UTP treatment in infected cells, in fact macrophages, can increase the release of ATP. Then we followed this hypothesis, considering the pannexin-1, the pore that Mariusz presented in his talk, and using carboxolone, that the pre-treatment with carboxolone, that is antagonist of pannexin-1, we can see that the UTP effect can completely be have a reversed, blocked. And also, if you use a macrophage, in fact, a macrophage from pannexin-1 knockout mice, UTP do nothing. So, suggesting that UTP can induce the release of ATP in a dependent way, in a dependence of opening of a pore, pannexin-1 pore. But what is the relation between these ATP and UTP on macrophage? So then we checked the possibility, the existence of one interplay between P2Y receptor, P2Y2 receptor, and P2X7 receptors. And we pre-treated cells, macrophages, with a select antagonist of P2X7 receptors. And we can see that this pre-treatment completely block the UTP effect, also blocked the ATP effect in this small concentration of ATP. And when you use macrophage from P2X7 knockout mice, we can see that UTP cannot induce reduction of parasite load, neither UTP, neither ATP. Establish a correlation between P2Y2 and P2X7 in this important control of the parasite. We also confirmed the LTB4 contribution using macrophage from 5-lipoxygenase knockouts. So as we can see here, in 5-lipoxygenase knockouts, ATP or UTP cannot reduce the parasite load. And also, we found that UTP in wild type UTP can induce LTB4 secretion as ATP, and in infected and uninfected, but UTP cannot induce secretion of LTB4 in macrophage from uninfected and infected macrophage from P2X7 knockout 1. Then we checked the cascade that involve caspase 1. So the requirement of caspase 1. And to do that, first of all, we use a blocker, an antagonist of caspase 1. And we see that pre-treatment with a blocker of caspase 1, we block the UTP effect. And also, if we infect the macrophage from double knockout, caspase 1 and caspase 11, and treat with UTP, you don't see anymore the reduction of parasite load. Then we check if the receptor for IL-1 is important for this effect observed for UTP using IL-1RA. it's the antagonist of, it's a blocker of receptors. And as you can see here, they use the pretreatment with IL-1RA, completely block the ATP in small concentration and UTP effect. Suggests that we need the IL-1 receptors to UTP being able to activate P2Y2 and have the effect. So finally, we treated mice infect mice and follow the development of the disease. And during the development of the disease, we treated with UTP on the prey. And we saw that the treatment was enough to reduce the swelling during the process. What was not observed when we do the same in animals, double knockouts for caspase-1 and caspase-11. And we found that after treatment, we can reduce the parasite load in the pay or in the drained lymph nodes. This was not observed in caspase-1 and 11 knockout mice. And we have the reduction of IL-1 beta, not observed in caspase-1 and caspase-11. So to make short, we can say that P2Y2 can activate panexin, open the panexin pore, and release ATP to extracellular medium that turn to activate P2X7 receptors that release LTP4 via each receptor, release IL-1 beta, and then we have the control of the parasite. To take home message, we can say that both P2X7 receptors and P2Y2 receptors can participate in immune response against Leishman infection. The interaction between purinergic receptors can shape the immune response to contribute to host resistance to Leishmaniasis. So the purinergic signaling might be considered as a good target for development of new drugs to deal with Leishmania-Amazonensis infection. I'd like to thank the group of the laboratory that we have been working many years in these projects, the collaborator from Federal University of Rio de Janeiro, and the collaborator from the other institutions in Brazil and abroad. I'd like to thank my group, different editions during these 11 years. And of course, I would like to thank you all for your attention.
Robson, thank you very much for this exciting talk, showing your great research in this area and also your contribution to the field. I will present shortly myself, yes, as the last speaker. So I'm working together now in the Brazilian Brewing Club with Professor Robson, with Ana Badascini, Ana Ventura, and new members of the Brewing Club on the organization of congresses, and also we are looking into how to disperse scientific knowledge of Brazilian researchers into to the international society, creating new collaborations, exchange, and so on. So I was the president of the Brazilian Brewing Club for 6 years. I'm counselor. I'm professor at the Institute of Chemistry, and I'm working in purinergic signaling on neurogenesis and neurodegenerative diseases. So I would like to go now to my presentation. My talk today will be on purinergic signaling and also on calcium signaling in neural stem cells, and what I will show, which are the mechanism and how these neural stem cells can be differentiated into neurons of a specific phenotype, such as, for example, GABAergic neurons. My laboratory, since the last 2 decades, is working on mechanisms how stem cells can convert into differentiated neurons and how we can form a complex network. And so we're looking on neurogenesis involving purinergic signaling and also involving calcium signaling. For this study, what I will show you, we used a mouse embryonic stem cell line. And this embryonic stem cells, basically they are isolated from the blastocyst stage, and upon specific protocols, they differentiate into cells of the 3 germlines, and they all including also neural cells. What I show here, using this mouse embryonic stem cell line, which we differentiate into neurons and glial cells through a protocol of 16 days. And what we can see here, that they are an excellent model for studying neurodevelopment bodies. They are growing out neural networks, and which give rise to differentiated neurons and also glial cells. In this case, shows here for neurons. What we can see here is that this differentiation is accompanied by increases in intracellular calcium concentration. And you see this here, but we have if the cells are induced to differentiation, then they transiently increase their concentration of intracellular calcium, as we can see here by an increase of fluorescence of a calcium-sensitive dye. Now is a question. So we have the participation of calcium. So we are very well known that we have these transients, we have these oscillations and waves. And now was the question: how would calcium channel be expressed and also participate at the event of neurogenesis? And what you can see here, that we have a differential expression regulation of L-type calcium channels, which are in the beginning upregulated. So it means that these calcium transients are needed to induce neural differentiation. And we can also see here that stimulation of voltage-gated calcium channels, that they are actually important for the triggering of neurogenesis. Here on the left side of this, we can see that there are voltage-gated L-type calcium channels expressed when we use KCl for depolarizing differentiating cells, when we can see that this response can be inhibited by a blocker of this L-type voltage-gated calcium channel. And now, okay, we have established the functions of the calcium channels in neurogenesis. Now I'm coming to this. You see that the influx of guided by spike activity in the presence of ATP. Yes, so the higher the ATP component becomes, so the lower basically is here the number. Yes, and what we can see that in the beginning there is high ATP concentration extracellular, and when differentiation goes on, this ATP extracellular concentration declines. And now we have made some hypotheses. I wanted to show you here, and when I show you the data, what may happen to induce neuronal differentiation. So, extracellular calcium enters through voltage-gated ion channels and also through purinergic receptors, which may be P2Y or P2X receptors, which induce intracellular spike and wave activities, where the wave activity comes from a release of intracellular calcium, which then finally act on the activation of neural transcription factor expression, and which would be here MESH1 and neurogenin 2. Yes? And these neural transcription factors, they have been shown to be involved in triggering of neuronal differentiation fate. So it shows here again, so that we have a downregulation of ATP when later stages of differentiation, but in the beginning ATP concentration is high. And what we see on the right side, that the embryoid body, which would go into differentiation, have high intracellular calcium concentration. And based on what we want now to show, we have postulated that there would be a neural clock which triggering calcium. Calcium wave and calcium spike transients, as well as oscillations in neural transcription factor expression. Yes? That means that we have purinergic receptors, that we have spike and wave activity, and that this would induce transient expression of neural transcription factors, such as, for example, MESH1, and this would trigger neurogenesis. And for this was made a transgenic mouse, yes, so expressing luminescence per seconds, or in seconds 73122 would block the liberation of intracellular calcium. And based on these studies, now we have identified receptors which are involved in this, so which are in the beginning of there are P2X7 receptors which are needed to maintain pluripotencies, and then the cells form embryonic bodies and differentiate. We could show that we need an upregulation of the P2I2 receptor, while on the same time, if we want to differentiate into neurons, we need to have a downregulation of the P2X7 receptor. And now we saw also that the P2X7 and P2I2 receptors, they are upregulated in undifferentiated cells in their expression, and we can also show that these two receptors were differentially expressed during differentiation of undifferentiated cells to NPC, to neural progenitor cells, and to differentiated neurons. And now we show the result of this calcium neural transcription factor imaging. Yes, we show it in top here the control, the calcium oscillations. Yes, and then we looked at MESH1. Neural transcription factor, and we see that we have a transient induction of expression, yes, and this is basically changed in the inhibition by inhibition of isradibine, and most importantly, a P2I2 receptor agonist such as 2S-UTP actually gives an immediate increase in MASH1 expression, a stable expression, yes, and this stable expression is consistent with an induction into ongoing neuronal differentiation. Yes? Any interference with intracellular calcium signaling by inhibiting intracellular stores or EGTA entering extracellular stores abolishes MASH1 expression. On the other side, we are looking for a neurogenin 2 expression, and there we could see that BC-ATP, which in these cells activates principally the P2X7 receptor, gives an immediate upregulation in expression of this neurotranscription factor. Yes? 2S-UTP, for example, does not have any effect on this. And now, so the question is, what is about GABAergic differentiation of embryonic stem cells? Yes, because this would be a defined neuronal phenotype, which would be important also for certain neurodegenerative diseases, such as, for example, Huntington disease. Yeah? Well, we can see here that beginning from the neuroprogenitor cell stage, yes, we have expression of GABAergic markers, which then increases when we have differentiated neurons. And now we are looking actually what happens if we treat, for example, with 2S-UTP, which in mouse cells activates the P2Y2 receptor. We see that we have a downregulation of most of the neuronal phenotypes, such as glutamatergic, dopaminergic, cholinergic, serotonergic, but we have an increase in the expression of GABAergic markers. And we are overall, that this treatment increases the number of GABAergic cells and GABAergic differentiation. Now we were looking at what is the ratio of MASH1 and neurogenin 2, and this ratio has been shown to be determined for many differentiation fates. First of all, you could see with BC-ATP that principally activates neurogenin expression will put down this ratio of between MASH1 and neurogenin 2. But on the other side, as expected, 2S-UTP activating P2Y2 receptor would upregulate the ratio of MASH1 divided by neurogenin 2. And based on this data, we could define that if we stimulate MASH1 on the cost of neurogenin 2, yes, then we would transform an oscillatory pattern which maintains the cells at the neuroprogenitor cell stage and also triggers differentiation into astrocytes. If you put this into a stable expression pattern and a high ratio of MASH1 over neurogenin 2, we will have basically GABAergic differentiation. Yes. So it means that high MASH1 ratios are important for the ongoing differentiation, while neurogenin 2 would be more important for maintaining the neuroblast stage. Yes, and what are the important players here for triggering GABAergic differentiations is the P2Y2 receptor as well as voltage-gated calcium channels. And just to put a conclusion, yes. So in this study, we looked at P2Y2 and P2X7 receptors and how they affect calcium waves and spikes patterns, and as well and how basically this receptor act on neural transcription factor expression, looking here at two, it is MESH1 and neurogenin 2, and the upregulation of the MESH1 expression would favor GABAergic fate determination of differentiating embryonic stem cells. Yes? And of course, there's also, together with these purinergic receptors, which in this case is a metabotropic purinergic receptor liberating intracellular calcium, we have the activation of L-type calcium channels, which are important for triggering neurogenesis. Yes? And overall, P2Y2 receptor activation could act as a therapeutic strategy. We'll have tomorrow another symposium where one of my postdocs will speak and show which are the implications of this study for Huntington disease. And finally, I would like to thank my co-workers, and principally who has done this work was my postdoc, Talita Glaser, with the help of other postdocs and students of my lab. And part of this work has been done in the lab of Ryuichiro Kageyama, who has this transgenic mouse models for transcription factor oscillations. And also Talita had an internship at the lab of Yang Teng at Harvard Medical School. And I would like to thank my local collaborators, Anna Maria Balestrini, Robson, Coutinho, Silva, Claudiana Lamio, Rodrigo, and obviously also I would like to acknowledge the funding agencies.